Digestion and Absorption of Proteins by Gabriel Degenhardt. When I first started working out, I was told by many a person that I needed to increase my protein intake so that I could build my muscles. But I was curious, how is it actually digested? How does the protein that I eat turn into usable nutrients in my body? Well, what is a protein? Well, proteins are large macromolecules made up of long chains of amino acids connected by peptide bonds, and are the building blocks of many of the body's tissues, especially muscle. However, proteins are very abundant in our bodies, and they are also used in other things, like hormones, enzymes, antibodies, and the skin, hair, and nails. So there are two types of amino acids which make up protein. There are essential amino acids and non-essential amino acids. Essential amino acids cannot be made within our bodies, so we need to eat food in order to get them. Non-essential amino acids can be made in our bodies, so we don't need to eat food in order to get them. A good, high-quality source of protein contains a good balance of the essential amino acids. A protein with more essential amino acids is considered a higher quality protein than protein with fewer amino acids. Examples of good sources of protein can be found in things like meat, eggs, and many milk products. Let's move on to the actual digestion of proteins. Digestion begins in the mouth. Here the teeth tear and crush food, and this increases the surface area for later digestion. However, no enzymatic digestion of proteins occurs at this point. After swallowing, food travels down the esophagus and into the stomach. It is here that true chemical and enzymatic digestion starts. The stomach secretes two things: hydrochloric acid and pepsinogen. Hydrochloric acid denatures proteins, and this changes the structure, making the peptide bonds more accessible for later digestive enzymes to act upon. It also converts pepsinogen to pepsin, the active form. Pepsin then hydrolyzes peptide bonds, which means it attacks and breaks the bonds, and converts the large protein molecules into large polypeptides. These large polypeptides then leave the stomach through the pyloric sphincter and enter the small intestine. At the start of the small intestine, the pancreas secretes 3 proteases, which are enzymes that break down proteins. These proteases secreted by the pancreas are trypsinogen, chymotrypsinogen, and procarboxypeptidase. These are all inactive forms of enzymes that require activation before they can break down proteins. Now, the enterocytes, which are the cells that line the small intestine, secrete something called enterokinase. And enterokinase converts trypsinogen to trypsin. And trypsin is kind of a big deal because it has 4 functions. 1, it hydrolyzes peptide bonds. Remember, peptide bonds are the bonds between amino acids. 2, it converts trypsinogen to more trypsin. 3, it converts chymotrypsinogen to chymotrypsin. And 4, it converts procarboxypeptidase to carboxypeptidase, the active forms. So enterokinase and trypsin activate the pancreatic proteases and enable protein breakdown to proceed. Each of these now activated enzymes continues to break down the large polypeptides as they travel through the small intestine. It breaks them down into small polypeptides and di and tripeptides. Attached to the edge of the enterocytes are things called brush border enzymes. And these enzymes stick out from the enterocytes like the bristles from a brush. And they hydrolyze peptides to very small tripeptides and dipeptides and individual amino acids. This would be the last stage of protein digestion before absorption of the proteins through the intestines. The small polypeptides and free amino acids in the small intestine must first be absorbed into the enterocytes and then into the blood. To be absorbed, they must first pass the apical membrane of the enterocyte, which is the side that faces the intestine. And then they must pass through the basolateral membrane of the enterocyte, which is the side that faces blood vessels. Absorption into the blood from the enterocytes takes place in many ways, but there are two main ways that it occurs. The first and main way that absorption occurs is that tri- and dipeptides cross into the enterocytes through the hydrogen channels following the hydrogen protons. Within the enterocyte, they can be further broken down by intracellular peptidases into singular amino acids, or they can be actively transported into the blood. The second main way that absorption occurs is that the free amino acids will cross into the enterocytes following the sodium through the sodium ion channels. Once in the cell, free amino acids can be transported into the blood. It's interesting to note that free amino acids are actually absorbed less efficiently than di- and tripeptides. So it is actually better to eat whole proteins than it is to try to eat a serving of separated amino acids. However, you would probably never eat single amino acids unless you had to. They smell really bad, and anyone who's ever left protein in a shaker cup for any length of time might be familiar with the smell. After being absorbed, the small peptides and amino acids can travel to cells where they can be used to build and maintain the body's tissues. There are other factors which might affect protein absorption. For example, protein structure. Whey, a protein found in milk and commonly used in supplements, is considered a fast protein and is digested and absorbed very quickly. This may be due to its water solubility. In contrast, the other major protein in milk, casein, is considered a slow protein because it forms clumps in the stomach, causing it to be digested and absorbed more slowly. This brings us to a question to help apply what you may have learned so far. Why do you think that the clumping of casein in the stomach might slow down its digestion? Because of the clumping, there is less surface area for the HCl and the pepsin to act upon. So there's less denaturation of the protein and less availability of the peptide bonds so that the pepsin can break them down. Other food content might also affect the rate of absorption of your protein. A food that contains protein but also contains fat or fiber, for example, will be digested and absorbed more slowly. This is because fat and fiber slow the rate that food moves out of the stomach and into the intestines. However, did you know that the intestines can also develop adaptations to absorb more protein? For example, during pregnancy and times of high metabolic stress, the intestines can increase in surface area to absorb more protein and other nutrients. Or, if in the case of a high protein diet, the number of brush border aminopeptidases and membrane transporters on the enterocytes can increase. It's interesting to note that the amino acid glutamine is the primary energy source of the enterocytes instead of fat or carbohydrates, as is the case with most other cells in the body. So in review, mechanical digestion of proteins begins in the mouth. In the stomach, HCl denatures protein, and the trypsin begins breaking the peptide bonds holding the protein together. Once activated in the small intestine, trypsin, chymotrypsin, and carboxypeptidase break down peptide bonds, further reducing polypeptides. Then, brush border enzymes hydrolyze peptides to tri- and dipeptides and free amino acids. These small peptides and aminos are then absorbed into the enterocytes and then into the blood for use throughout the body.